Today we're going to continue our molecular biology unit and we are going to do our lesson 8 DNA technology. So take out your note taker for lesson 8 and get ready to take some notes. All right, here we go. So we're going to start with understanding a little bit the connection between science and technology. We know that they are tightly linked, and we know that technologies have improved our understanding of DNA, its function, and how its code can be manipulated for a variety of purposes. So we know that two technologies that we're going to talk about today, which is genetic engineering techniques and recombinant DNA technology, they would not be possible if it weren't for the advances of technology. So keep in mind that science and technology are tightly linked. There is a very strong connection, and as technology advances, so does the field of science. So we are going to start with the definition of recombinant DNA and how recombinant DNA is used in different technological ways. So recombinant DNA is made by taking segments of DNA from two different species. So it could be the human species and a bacteria species, and you can combine them into a host organism to produce new genetically modified combinations that are of value to science, medicine, agriculture, and industry. So in your notes, you're going to list some examples of how recombinant DNA technology can be used. So for example, under therapeutic products, you know that recombinant DNA technology can be used in the form of vaccines or antibodies or anti-cancer drugs. For genetically modified products, you know that we can use GMOs in the form of fruits and crops, animals, and they've all been made through recombinant DNA technology. For diagnosis, there's gene therapy, CRISPR, therapeutic strategies, and for energy applications, we can use recombinant DNA technology to make bioethanol or biomethanol, for example. So there are 7 tools and techniques that we're going to focus on, and these are examples that are used in genetic engineering. So in your notes, you're going to write down these 7 examples: restriction enzymes, DNA ligase, bacterial plasmids, polymerase chain reaction, gel electrophoresis, cloning, and CRISPR-Cas9. So there are 3 items you're going to need to recombine DNA. So the first one is restriction enzymes. And let me get my pointer here. So restriction enzymes are enzymes that cut or cleave DNA molecules at or near specific sequences. So these are the molecular scissors. So then you can cut a particular sequence of DNA here, and then you can glue them together through DNA ligase, which is another enzyme that pretty much seals the 2 recombinant DNA strands. And then you can take that recombinant DNA and you can introduce it into bacterial cells. So we know that bacterial cells have plasmids, which are small circular double-stranded DNA that naturally exist. So when the bacteria replicates and divide, they can take our recombinant DNA here in the form of these plasmids, and they can recombine them and make more clones of our desired recombinant DNA. So let's look at the 7 examples of the different techniques used in genetic engineering. So you're going to need to describe each genetic engineering technique and what it's used for. So polymerase chain reaction, or simplified as PCR, is used to rapidly make millions to billions of copies of a specific DNA sample. So you can take a very small sample of DNA and then you can make a lot of it. And then you can take that DNA and you can use it for cloning, mutation detection, sequencing, microarrays, or you're probably more aware of how it's used in forensics or paternity testing. You don't need to know all the details, but PCR requires a machine known as a thermal cycler. It requires an enzyme known as Taq polymerase that can withstand really high temperatures so that when you denature the DNA or you separate the strands, notice that the enzyme is going to be able to withstand temperatures as high as 95 degrees Celsius. Okay. You're going to need your DNA sample that you want to make copies of, and then you're going to need primers so that the DNA know where to start the annealing process or making the extensions of the DNA that it wants to copy. And you're all going to stick that into a tiny little PCR tube, and you're going to run about 30 cycles depending on the size of and the condition of the thermal cycler, how new it is, and you're going to end up with billions of copies of a tiny little piece of your DNA sample. So what can you do with that DNA that you've amplified now? Well, there's another technique known as gel electrophoresis, and this is used to separate DNA fragments according to their size. So what you do is you take a machine known as a gel electrophoresis machine, and you're going to prepare these gels made out of agarose, and you're going to apply these combs to one side of the gel so they make these indentations known as wells, and that's where you're going to add the DNA that you want to separate. So we know that DNA is negatively charged, so when the DNA is broken up into tiny little pieces by adding different restriction enzymes, that DNA is going to travel to the positive side. So this machine here is going to apply a voltage to the gel, and one side is going to have a negative voltage and the other side is going to have a positive voltage. So the DNA is going to travel from the negative side to the positive side according to its size. So then the larger, heavier pieces, here you see this piece about 1,000 BP bases, it's going to be very slow, and it's probably not going to be able to travel as fast as those that are shorter, maybe 100 kilobases here. Okay, so then what you end up with is a DNA fingerprint, and you can use this fingerprint to investigate crime scenes, to analyze results of PCR reactions, to analyze genes associated with a particular illness, or for DNA profiling, to study different species in a taxonomic study or in paternity testing using the DNA fingerprint that you see here. So gel electrophoresis results give a DNA profile. So a DNA profile is like a fingerprint. And notice that even though we are all unique, most of our DNA is actually identical to other people's. But we have these tiny little specific regions that are called polymorphic regions. And these regions vary among people. And we inherit these different combinations from our parents. So then we can take these polymorphic regions, which are those tiny variable regions, and we can analyze to give our own unique DNA profile. So the DNA profile that is made at the end of a gel electrophoresis process is going to be our own DNA fingerprint that is going to be just like our thumbprint, right? Is going to be unique to us. So then we can take that DNA profile or DNA fingerprint, and we can, one, determine paternity, because we know that the child has to have the DNA of both its parents combined. And we also can study crime scenes, because the suspect has to have the exact DNA profile as the one at the crime scene. So here, notice that this is a result of a DNA profile. So this is how the DNA fragments have separated into tiny little pieces, those polymorphic regions. And we see that if this is, let's say, the DNA found in blood in a crime scene, or the DNA found in hair in a crime scene, then you can match this DNA to different suspects. And notice here that suspect number 2 is a perfect match. That means that this region of DNA matches exactly to this region, and this region, and this region, and this region, and so on. So how do we read a DNA profile when it comes to a paternity test? So we know that this child has to have a combination of DNA of mother and child. So we first see which bands of DNA match mom. You see that here for this child, this band matches mom, and this band matches mom, and then this band matches mom. So we're left with the other bands, like this one, and we have to match it with a particular dad. So we know that there is no match here with dad 1, so we can exclude dad 1. We see there's a match here with dad 2 and dad 3. So one of these two guys happen to be the father of the child. So then we move on to the next region that does not match mom. So these two regions right here, and we see that these two regions perfectly match dad 3. So we can say with about 99.99% certainty that dad number 3 is the biological father of this child. And we know that these DNA fingerprints or profiles are acceptable in court. All right, what is cloning? Cloning is a technique scientists use to make exact genetic copies of living things. We can make copies of genes, cells, tissues, and even whole organisms can be cloned. We know that nature can already make clones of their own. For example, bacteria can make exact copies of themselves. Also, identical twins are considered clones. And we know that there are different types of artificial cloning, known as gene cloning, reproductive cloning, and therapeutic cloning. So here you can skim through a cloning timeline, and you can see the different animals that were cloned throughout the years. Even though there was cloning as early as the 1950s, we know that the first whole mammal was cloned in 1997, and this was a sheep named Dolly, and it was the first mammal to be cloned, and of course it brought in a whole public debate about the prospects for cloning humans and if that was even possible. So we know that a wide array of mammals have been cloned in the past. And here we see some examples of some other mammals that have recently been cloned, including a project that it's still in progress of the cloning of a woolly mammoth. So we don't know if scientists will be successful with this, but it's in progress. So the process of cloning is pretty straightforward. You don't need to know all the details of the process, but let's say you want to clone this adult here in orange. Okay, you want to make an exact copy of this adult. Well, what you need to do is take some cells from this adult. It could be skin cells that have DNA, and you're going to need an egg that is donated from another adult female of the same species. And you take that unfertilized egg and you take out all of its DNA. So here, this in blue, you remove all that. Well, not in the dark blue, right? You remove all that, and now the cell is hollow completely. So you take that hollow cell and you fuse it with the cell of the adult that you want to clone. And when the cells fuse and start dividing, you get an early stage embryo here. And then you can take that embryo and you can implant it into a surrogate mother. So that means that's the mother that's going to hold the embryo and give birth to it. When this mother gives birth to it, this cloned animal here is not the biological baby of the surrogate mother. It is actually a clone of this adult here that you were trying to clone from the first place. So this adult up here is 100% identical DNA-wise to the cloned animal here. Okay, so it's a pretty straightforward process if you think about it. All right, moving on. So what is CRISPR-Cas9? So we talked about this a little bit before. CRISPR-Cas9 is a DNA editing tool. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. And then it has a CRISPR-associated protein 9. And we know that this is a very exciting time that we are living in right now because we have this new tool that's generated a lot of excitement in the scientific community. It is fast, it is cheap, it is more accurate, and it's more efficient than other existing genome editing methods. So the way that this tool works is pretty much like a find and replace function in your word processor. So you take a particular section of DNA that perhaps it's damaged or mutated, and you take the guide molecule, which is a specifically designed molecule of DNA, and you associate it with the Cas9 protein. And then what it does, it finds this defective DNA and it cuts it. And then your DNA is then able to use its own machinery to make or replace it with a healthy copy of itself. So scientists are still working to determine all the details of how this works and if it's safe and effective, but it's still something that is very exciting because it has a wide array of possibilities. So scientists think they can treat diseases like cystic fibrosis, hemophilia, sickle cell anemia because they are single gene disorders, right? What if we can just find that gene and cut it out and replace it with a good gene? So this would be a big deal, right? And it holds a lot of promise for treatment and prevention of more complex diseases like cancer, heart disease, mental illness, and HIV. So we'll see. We'll see what happens. If you want to know more detail about the CRISPR-Cas system, you can read through this. Again, you're not responsible for knowing all of the details, only that it's a DNA editing system that can cut and replace a erroneous or a defected segment of DNA. All right, moving on. So in your notes, you're asked to list 3 specific examples that show the benefits of genetic engineering. So the 3 examples that you are to know are human growth hormone, insulin, and pest disease resistant fruits and veggies. So all of these products are made through genetic engineering. So human growth hormone is used to treat growth disorders in children. So you can make, if you want to call it synthetic, genetically engineered human growth hormone by recombining the human gene for growth into a bacterial plasmid. And then that bacterial plasmid will make millions of copies of itself, and then you have a fast and efficient way of producing the hormone. Before this, notice that the only way that the human growth hormone could be produced was a laborious process that you had to isolate the hormone from the pituitary gland tissue in humans, and this was done in cadavers, and it wasn't very efficient. So now we can do this synthetically, and it's a great benefit. Insulin used to treat diabetes is pretty much done the same way. You take the human gene for diabetes, for the human gene for insulin, take the human gene for insulin, and you recombine it with the bacterial plasmid. And again, you can make hundreds of thousands of copies of the insulin hormone that you can isolate, and then you can use it to treat humans with diabetes, which is high blood sugar. You can also make genetically modified produce by genetically modifying crop plants. And by doing this, you can make plants that are more resistant to certain pests or tolerant to a particular type of herbicide, or maybe tolerant to a particular location, like if it's too salty, or also tolerant to high temperatures or low temperatures. So you'd be surprised if you click on this list of bioengineered foods, you would see that a lot of the foods that we find in the supermarket that are non-organic foods have been genetically modified one way or another. So 95% of all the soybean, corn, sugar beets, canola, and cotton and apples are genetically modified products. All right, so the last thing you are going to want to write down in your notes are some pros and some cons of genetically modified organisms. So what's good about it, what's bad about it. You can click on each link to learn more. So we know that GMOs are easier to cultivate. They boost the yields that an individual can generate on existing lands. They can be transported to inaccessible regions of the world. You can preserve energy, soil, and water using GMOs. But then some cons could be that maybe some people might be allergic to some of these genetically modified foods. There is a link to how these GMOs can contribute to antibiotic resistance, to maybe a link to cancer or herbicide resistance. So take a look at some of these pros and cons and make sure you write some of them in your notes. And that's it for today. Hope you enjoyed your lesson.